Okay, we've talked about primary and secondary structure. Now we're going to get into tertiary structure. Now, tertiary structure, remember, is the overall structure of a polypeptide. What's its general shape? And sometimes the general shape is the combination of secondary structures like alpha helices and beta sheets. How are they arranged in a polypeptide to give an overall shape for the protein? Now, sometimes the tertiary structure is mostly dominated by a particular shape, and those types of proteins are called fibrous proteins. And we'll show keratin and collagen as an example. They have relatively simple tertiary structures, and that simplicity usually means that fibrous proteins are structural proteins. They're there as building blocks for cellular structure. You really need the complexity of a globular protein, one that has alpha helices, beta sheets, all sorts of stuff in it, in order to make things like enzymes, which have really complicated functions. But let's start with fibrous proteins, and let's start with keratin as an example. Keratin is a structural protein that's present in your hair, skin, nails, etc. Some of you may have had keratin treatments on your hair. And when we think about keratin as a structural protein, it's based on a simple motif of a coiled coil structure. So there are two polypeptides that sort of bind to each other in this coiled coil. Here's a good example of it. And so the simplest unit here is a dimer. Okay, these polypeptides wrap around each other, and the reason that they do that is because of hydrophobic interactions. If you look at each of the monomers in this dimer, there is one side of these polypeptides that have hydrophobic residues. And so if you have a monomer like this with a side of hydrophobic, another monomer like this with a side that's hydrophobic, and you put them in a hydrophilic solvent like water, what happens is that they sort of coil around each other to hide this hydrophobicity in the interior of the dimer, right? Leaving the hydrophilic residues on the outside to interact with solvent. So this coiled coil is primarily formed just through hydrophobic interactions on the interior of the dimer. Now, this dimer can form larger structures by having different dimers bind to each other to form protofilaments. These protofilaments can bind to other protofilaments to form microfibers, and eventually you get big fibers of keratin. Now, although the dimer is formed by hydrophobic interactions, the larger structures here between dimers and so on and so forth are formed through cysteine disulfide bonds. So one dimer can bind to another through disulfide bonds, and so there are some cysteine residues in the keratin polypeptide that's responsible for these higher order forms of organization. But the coiled coil is the basic motif, and that's why we think of it as a fibrous protein. Let me show you a picture of keratin, if I can get out of this. And here's keratin here. Here's that coiled coil structure of keratin. And if we rotate it around, we can see how each of these monomers sort of wraps around the other. In red here, I've got cysteine amino acids highlighted. And so those are sort of displayed on the outside of this coiled coil so that one dimer can bind to another dimer. And let's look at one other thing to highlight here. Let's see the hydrophobic residues. If I just show you hydrophobic residues, you can see that they're sort of buried on the interior of this coiled coil, hiding as much as they can from the solvent. The hydrophilic ones, as shown in the green and the light blue here, are interacting with the solvent. Okay, so that's keratin as a fibrous protein, very structural and very really kind of simple to describe its tertiary structure. So let's show another example, right? Collagen is another example, classic example of a fibrous protein. This is one of the major connective tissue structural proteins that are in your bones. It's in your cartilage, things like that. And there's some diseases of collagen deficiency that we'll talk about in class that have real terrible impact on bone and cartilage structure. Now, if we think about what is the structure of collagen, it is based on a triple helix structure. So here's a good picture of it on the right side of the slide. We've got 3 polypeptides that sort of wrap around each other into a triple helix, very different than the keratin. And if we look at the amino acids that are present in the polypeptide of collagen, we can see that a lot of them are glycine, some of them are proline, and some of them are hydroxyproline. Now, if we think of those amino acids, proline is hydrophobic, glycine is kind of hydrophobic, it's kind of innocuous, right? Hydroxyproline is definitely not hydrophobic, right? It's got an alcohol group on proline. So one question is, where did hydroxyproline come from? That was not one of the 20 naturally occurring amino acids. And so this is an example of post-translational modification. Glycine, proline is coded for, and then enzymes come in and recognize that second proline in the these amino acid patterns and modifies it through hydroxylation. So this is an example of post-translational modification of a protein. And the presence of these alcohol groups should make you think that this triple helix probably does not associate like keratin did through hydrophobic interactions. And that's true. The triple helix uses these hydroxy groups to assemble this triple helix using hydrogen bonding, right? So let's look and see how these monomers, or these peptides of the triple helix, sort of interact with each other. Here's our glycine, here's our proline. Remember, that's the amino acid that causes the bend. And here's our hydroxyproline with the alcohol group here. This alcohol group can share electrons with hydrogens from a neighboring polypeptide through hydrogen bonding to stabilize this triple helix and with these 3 chains. Here's electron microscopy image of collagen. And so just like keratin, where that dimer can associate to higher order structures, the triple helix of collagen can assemble with other triple helices to form these structural cables that sort of tie together your bones and your cartilage, giving it rigidity. Now let's look at the collagen, show an image of collagen here. Here's our keratin, here's collagen, and we'll see some images in class of this too. It's kind of hard to see this triple helix, but if I rotate it around, you can maybe follow one polypeptide, a second polypeptide, and then a third polypeptide behind there in these triple helices that are curling around each other. And inside the triple helices are the hydrogen bonds that are occurring between the hydroxy groups of the hydroxyproline and the hydrogen from another chain.